Hi, my name is Magali Soumillon, and I'm the co-founder and CEO of Flexomics. In this short presentation, I will introduce the Flexomics platform, a technology designed to connect live cell functional imaging with single-cell sequencing. The core of the technology is contained in this consumable that I have here in my hand. Our goal is to enable experiments where researchers can observe how individual cells behave and then directly connect those behaviors with molecular information, such as gene expression or immune repertoire sequences. This work has been supported by NIH SBIR funding, both from NHGRI and the NCI. Let me start by briefly explaining the gap we are trying to address with this technology. On one side, imaging and flow-based methods allow researchers to observe phenotypic responses at the single-cell level. We're talking about cytokine secretion, morphology, or even cell-to-cell interaction. On the other side, single-cell RNA sequencing gives us that depth that allows us to define transcriptional states and molecular identities across many cells. However, these two measurements are very rarely linked from the same individual cells. The Flexomics technology has been designed to integrate these measurements within a single experimental workflow. And again, this is all happening in this consumable. First, single cells are captured on a slide-sized consumable containing an array of picowells. Cells can then be cultured on chip and exposed to different types of reagents for functional assay and phenotypic measurement. Standard fluorescence microscopy is used to capture that phenotypic measurements from individual wells and individual cells. Following imaging, RNA from those same cells is captured and sequenced, and I'm going to tell you more about that. Because each well is indexed, sequencing reads can be linked back to the cells that were previously imaged, generating integrated data sets combining phenotypic information and gene expression. How does it work? The Flexomics platform relies on optically encoded capture bits that are co-loaded with cells in that picowell array. Each picowell contains a barcoded bit that is designed for capturing mRNA molecules. We're talking oligo-DT and unique molecular identifiers to enable like single-cell RNA-seq workflows. And that decoding step really allows us to map out where those barcodes are on the consumable. This is part of our manufacturing strategy. After sequencing, the bit barcodes allow us to link back those sequencing reads to a specific well and so to a specific cell that was previously imaged. This is what allows us really to enable that direct integration of cell phenotype and gene expression from the same cells. How does it look like to work with our consumable? It is really easy to use, and I'm going to show you how that looks like. Again, we load the cells, we capture, we do the assay, and we do the imaging. All of that is happening on that one consumable. To load cells and exchange reagents, we just use traditional pipetting. We designed the consumable to be compatible with multi-channel and liquid handler. To keep cells alive, we basically just incubate them in our CO2 incubator. And again, there's no instrument here. Basically use your current infrastructure and your current microscope to look at cells in the device. We designed this to really make it easy to use. So we have those layouts of many 384 well plates in a way. It's like 16 by 24. We have one fiduciary mark per those 384 wells. So it makes it very easy to know where we are on the consumable. This is how it looks like when you look at fluorescent data. You really have like mapped those cells and those responses. This architecture really enables new types of experimental format that are usually pretty difficult to do in droplet-based single-cell workflows. For example, we can do cell-to-cell interaction where we co-load immune cells and antigen-presenting cells. I'm going to tell you more about that. We can do cytokine secretion. We can do a bunch of sequential assays, looking at activation markers prior to doing this RNA capture and sequencing workflow. We've also shown that we can also do like adherent cells on that system. So it means that we can basically seed the cells, let them attach, treat them with drugs, and then do the classical staining that people use for like toxicity assays and things like that directly on the consumable. In general, this system is really designed to combine that live cell functional imaging prior to this molecular capture. Let's talk about data. So here we're talking about two different types of data. We're talking imaging data and sequencing data. So to help with the analysis, we develop a UI UX that really allows us to connect those two types of data together. So basically, we can navigate all the imaging data. We can pick specific wells where we see something interesting. We integrated all the classical single-cell RNA-seq analysis that you would traditionally do. The big difference here is we can literally click on a dot in the UMAP and see the image of that specific cell. We can look at specific genes, we can do differential gene expression, and we can export really those analyses at any time for further analysis downstream. Again, looking at gene expression data, downregulated, upregulated genes, looking at a heat map, we look at like specific wells and specific responses, and then we do across the population or across like a specific subset of wells. From the imaging data, we can do the same. We can actually basically look at like all that data and really like plot this and map it out to be able to look at specific wells and specific responses we're interested in. It looks a bit more like flow cytometry. We're talking about like being able to gate and look at specific cells with specific fluorescence responses and really track them across the entire analysis, both on the imaging and on the sequencing data. So we can gauge specific population. And again, we can really click on the dot and see the image of that specific cell. And at the end, we can really integrate this. We can basically layer that fluorescent phenotypic data on top of, let's say, UMAP created with sequencing data. So very powerful tool to really integrate two different types of data. After that, I just wanted to give you a few examples of the type of experiments you can do with this platform. So here, this is an example where we do like kind of like pretty complex workflow, which is like looking at immune response in a cell-to-cell culture system. So here we have antigen-presenting cells that are primed with peptide, and we co-culture them with CD8 plus T cells. On the next day, we measure functional response doing cytokine secretion assays and looking at also a few other activation markers to see which T cell got activated by that specific antigen. Once we're done with the imaging, again, we lyse the cells, capture the RNA for sequencing. And here what we did was basically doing both like RNA sequencing on a specific T cell panel and looking also at T cell receptor to do a clonotype analysis. It really shows you like the type of data you can really like bundle together. Here we're talking like functional response by imaging plus gene expression plus T cell clonotype. So type of complex assay you can do on this consumable. The other example I wanted to show you is how we do this for like adherent cells. So again, here we're looking at like seeding the cells, letting them attach, treating them with a drug, doing like more complex staining, and then again doing harnessing and combining that data. This example here is A549 blood cancer cells that we treated with etoposide directly on chip, and we imaged them on the next day and did also like the single cell RNA-seq on that population. Really, this enables the transcriptional responses to be directly linked to the phenotypes observed during imaging at single cell resolution. NIH SBIR funding has been critical for the development of the Flexomics technology. Early work was funded by the NCI to look at feasibility around immuno-oncology application and the integration of T-cell receptor sequencing. Subsequent support from NHGRI really enabled us to expand on the consumable architecture, refine our barcoding system, as well as develop this software infrastructure I just presented, and help us also like start to get going on like the automation of the workflow. This support also enabled us in deploying this platform to early access users, and we've learned a lot by working with others. And so really, we're really grateful and very fortunate to have gotten that funding from NIH. Looking ahead, we are continuing to expand both the technical capabilities and the deployment of the platform. This includes new workflows for functional immune repertoire profiling, higher throughput screening formats, and expanding on molecular readouts. We're also working with early access collaborators and feasibility partners to integrate the technology into functional genomic workflows. If you are interested in exploring how this platform could support your research, we would love to talk to you, so please don't hesitate to reach out. And thank you for your time and for your interest in Flexomics.